Hello, everyone. Myself and Dr. Arna Christensen have been working with Daphnia and investigating the influence of microplastic color and odor on feeding preference in these animals. And Daphnia are freshwater crustaceans. You can see a close-up of one here, and some Daphnia feeding on some microplastics on the left. Just to give you some perspective, Daphnia are about the size of a grain of rice, so they aren't quite microscopic, but they are quite small. And I also mentioned we were working with microplastics. And microplastics are small pieces of plastic classified as less than 5 millimeters in diameter. And we see microplastics everywhere. They're in the ocean, they're in soil, they're at the tops of mountains, and even in our drinking water and in our air. Currently, the most common type of microplastics in aquatic ecosystems are microfibers. And these are microplastics that come from clothing made of synthetic textiles like polyester. And when you wash them, they shed microfibers that enter the water systems and eventually aquatic ecosystems. So these are the types of microplastics that we use for our experiments. One of the concerns with microplastics is that small organisms like Daphnia are eating them. As these small organisms eat microplastics, there are concerns of bioaccumulation, which occurs as larger organisms eat these small organisms and the microplastics accumulate in larger predators. There are also similar concerns of biomagnification, which involves the toxicity of the chemicals these plastics are made with or absorb, becoming more potent as they move up the food chain. So microplastics are directly being consumed by small organisms, but still making their way up into top-level predators, including us. For our experiments, the first thing we needed to do was determine how we could analyze these microplastics. So you can see here some yellow microplastics I prepared from a swatch of polyester fabric we purchased from Joann's. When we took a look at our microplastics under a microscope, we were initially worried about determining fiber color just from these bright field photos here, because the color of the fiber gets washed out a bit. The black and the red fibers were able to be identified with these bright field images, but our green and our blue microplastics look quite similar. However, it turns out that the microplastic colors that we were using had unique autofluorescent properties. And this was great news because it gave us a mechanism to identify fibers with a high degree of confidence. So you can see here all of our colored fibers are autofluorescing red, but only our green fibers here autofluoresce under the green emission. And this was key because it allowed us to distinguish our green microplastics from our blue. So now, having a way to analyze our future data, we set off to conduct some experiments. However, I wasn't quite done dealing with fiber preparation yet because I still needed to make enough microplastics for the experiments we were going to conduct. We realized that the best way to prepare fibers was to cut up a large batch of each color and then weigh out a predetermined amount and add them to combo media to create a stock solution that we could easily pipette into our treatment jars. And that stock solution for each of our colors you can see here. We also had a constant Daphnia culture, so we had Daphnia on hand when we needed adults for experiments. But before Daphnia were exposed to microplastics, they were fasted for 24 hours to make sure they would feed on our microplastics and their intestines weren't too opaque. Then we had two main sets of experiments, one looking at microplastic color and the other looking at odor, and these were done independently. But for both experiments, Daphnia were exposed to microplastics, then the intestines were dissected and imaged, the fibers were counted using those autofluorescent properties I talked about, and we went through and analyzed our data. Our first set of experiments looked at color preference, and my hypothesis here was that Daphnia would prefer green microplastics because they would resemble their algal food source. Other studies have found that aquatic organisms preferred plastic that resembled their food, and an example you might be familiar with is sea turtles eating plastic bags because they look like jellyfish in the ocean. So we set up 3 treatments with a 50-50 ratio of green microplastics mixed with either black, blue, or red, and here you can see some of our Daphnia eating microplastics in an exposure jar. And each of these treatments contained 8 animals that were exposed to a final concentration of 0.01 milligrams per milliliter of microplastics for 1 hour. And again, these Daphnia were fasted for 24 hours prior, and then exposed, fixed, dissected, and then imaged. So here you can see one representative image of the dissected intestines. For each of these groups, we have our black and green, our blue and green, and our red and green mixed microplastics. And just a reminder that this red emission in the middle here is showing our green, our blue, and our red microplastics because they all autofluoresced red. But our green emission down at the bottom here is just showing those green fibers. And when we went through and analyzed our data, We didn't find a significant preference for color in any of our groups. There was a slight preference for black and for blue in our first two groups, and a slight preference here for green in our red and green group. However, this difference was not statistically significant. But things got a little bit more interesting when we moved into our odor experiments. This involved aging microplastics with algae to biofoul it. So we took some green and some black microplastics and aged it in combo media with algae or without algae for 3 weeks in our greenhouse. You can see our two groups here. And the microplastic that was aged with algae is referred to as fouled, and the microplastic aged without algae is referred to as pristine. And my hypothesis for this set of experiments was that Daphnia would prefer microplastics that had been biofouled because they would mimic the olfactory cues that algae normally secretes. We had 4 treatments with a mix of green and black, fouled, and pristine microplastics. And each of these treatments contained 8 animals that were fasted for 24 hours and then exposed to a final concentration of 0.01 milligrams per milliliter of microplastics for 1 hour. And again, these animals were then fixed, dissected, and imaged. Here you can see a figure of the dissected intestines for one representative animal from each of our groups. So we have our pristine microplastics here on the left. We have our fouled and pristine mixed microplastics in the middle. And then we have our foul green and our foul black microplastics here on the right. And in this case, because we were just using green and black fibers, we focused on this green emission at the bottom to count our green microplastics, and then our bright field images to count our black. And when we analyzed our microplastic counts, we see a clear preference across the board for black microplastics. And this difference is significant within all of the groups with biofouled microplastics, so these last 3 groups here. And when looking at the ratio data across groups, again, it's clear the preference for black, but we see this preference significantly heightened in the presence of these biofouled microplastics. And the thought is that it isn't actually the biofouled microplastics themselves that are attractive, but the fact that they're releasing olfactory cues into the water that are promoting feeding behavior in the Daphnia that's heightening this underlying preference for black that otherwise isn't significant. The preference for black is most intense when both sets of microplastic were biofouled in this group here. So we believe that the more olfactory cues in the water there are, the more heightened its preference for black will be. Lastly, if it was the individual pieces of plastic that were more attractive, we would see a preference for green microplastics in this group where we have fouled green and pristine black. But that's not the case. We're still seeing this preference for black here, indicating that it is the presence of these olfactory cues in the water that's triggering feeding behavior and heightening this preference for black microplastics. In summary, one of the most important findings was the autofluorescent properties of our microplastics. This could be really valuable for future studies and was instrumental in allowing us to analyze our data with a high degree of accuracy and confidence. As far as our results go, we found that microplastic color is not a significant factor in fiber preference unless olfactory cues are present. And when those olfactory cues that are normally given off by algae are present, we see this heightened preference for black microplastics over green. If we were able to continue our research, we would want to confirm that these olfactory cues are diffusing into the water from our microplastics. It would also be interesting to identify which chemicals are being secreted into the water and determine the threshold for olfactory cues to see this impact in preference. Lastly, it would be interesting to determine if our findings are true for other types of microplastics. We use microfibers for all of our tests, but other types of plastic vary in their ability to absorb and secrete chemicals, so we cannot assume that these results will hold true for other types of microplastics. Lastly, I would like to take a moment to thank Dr. Arne Christensen for taking on this project with me and for all of his help and support over the past year. I would also like to thank Dr. Jessica Stevens and Dr. Frank Giuliano for all of their feedback and advice during this process, and the Westfield State Biology Department for allowing me to use the labs and resources required for this research. And thank you for listening and learning a bit about the world of microplastics and Daphnia with me today.